Hey everybody, Mr. Lane is back. Today we're going to be talking about the most important thing in this entire semester. It is transcription and translation, or what we like to call protein synthesis. This whole idea of how do we go from DNA, how do we go from this awesome sauce molecule right here and make a protein. So how do we go from a genotype into a phenotype? So this is going to be a foundational understanding in all of biology. So pay close attention. Here we go. First, before we start, this wicked cool structure that we have right here is called a protein. This is a model of a very complex protein. So these protein structures are very, very intricate, and they are in 3 dimensions. The code that we have in DNA is what leads to that very unique 3-dimensional shape. Your learning targets. This is the big picture, and this is for the entire unit, not just this video. You will be able to say these things confidently and proudly, that I can explain how DNA is transcribed into mRNA, that keyword there is transcribed. The second one is I will describe how mRNA can be translated into a polypeptide. So again, translated. If you think translated, you're changing the language. And the third one, I can compare the structure and function of DNA and RNA, because as we know, structure equals function. And finally, I can explain how a mutation can affect a protein. We'll talk about those in class. The questions I want you to be able to answer right now. Maybe I should leave that part in. I'm just going to edit it out. All right, so your essential questions. How is DNA transcribed in RNA? By the end of this chapter, you need to be able to answer how is DNA transcribed into RNA using very specific detail. And how is RNA translated into a polypeptide? A polypeptide is like a baby protein. So those are those 2 big questions that you should be asking yourself. Can I answer these? Again, here's another wicked cool model of a protein. All right, we got to go all the way back to Plumpy-Nut, which is in semester 1, that awesome project that we all did. We have to talk about proteins and their structure. The monomer, the simplest unit of a protein, is called an amino acid. So a long chain of amino acids comes together to make a protein. A polypeptide is what we call that long string of amino acids. And remember, just break that word down. If you see the word poly, that means many. And peptide is the type of bond that we have between amino acids. We'll talk more about that in class. There are only 20 amino acids that make up all living things, or I should say 99.9% of all living things. In human bodies, there's only 20 amino acids. So think about that. Of all the different proteins that our body makes, there's only 20 different amino acids that make them. That's like saying there's only 20 different Lego bricks, and think of all the things you can make with those 20 different Lego bricks. And finally, those proteins are assembled on a ribosome. Remember this all the way back in the cell unit from semester 1. So proteins are assembled on a ribosome. Here is the big picture, aka the central dogma of biology. We are starting with DNA. DNA is like a reference book. It's stuck inside the library. In the cell, we call that library the nucleus. So DNA is simply this big book that we can open up and we can read it, but we can never take it out of the nucleus. But we need to get the information from it. So what are we going to do? We're going to transcribe the information that's in the DNA into the form of RNA. So this RNA is more or less a note that we open up the DNA and we write down what we see. We take that note of RNA and then we change the language. We translate it outside of the nucleus in the cytoplasm into a protein. Okay, so we're going with DNA, copying down the message in DNA in the form of RNA, leaving the nucleus, and then translating, changing the language from RNA into protein. This is essential to understand. So please, please, please pay attention. All right, this is where it gets a little fun. How do we compare DNA and RNA? They are very different. When we talk DNA, we know that DNA is double-stranded, that we have 1 strand and 2 strand. We know that it had that deoxyribose sugar right here, and that it had ATCG. Okay? RNA is slightly different. RNA is typically single-stranded, so it's not this double helix. It would be like what we call a single helix. It also has a ribose sugar. In fact, that's where the name comes from, ribonucleic acid, as compared to deoxyribonucleic acid in DNA. In RNA, there is no thymine. There is no T. Instead of T, we have U for uracil. Okay, that might be sound like kind of like some kind of childish name. You could call someone like, no, uracil, but that's not what it is. It's actually nucleic acid, base in the nucleic acid of RNA. The way I like to remember this, and I will be putting this out many times, there is no T in RNA. Or, as I like to remember it, Mr. T says, pity the fool who forgets about uracil. And if you don't know who Mr. T is, Google it real quick and you'll get my joke. But Mr. T says, pity the fool who forgets about uracil. There is no T in RNA. Okay? Both contain a sugar, a phosphate, and a base. Cool. Here's a diagram of RNA on the left. We can see that that's single-stranded. It's not double-stranded. And we can see the C, G, A, and U. And over here we have the double-stranded, the double helical structure of DNA. And again, C, G, A, and T. Now check this out. If you look at the structure of thymine over here and uracil over here, really the only difference is this methyl group, or what we that CH3 group here. So 4 atoms different between the two. That's kind of cool. 4 atoms different. You go from something that's very much DNA to something that is very much not DNA. And it's just 1 carbon and 3 hydrogens different. Okay, the last thing we're going to talk about today is the flavors of RNA. There are 4 major types of RNA. The first one is this mRNA. The M stands for messenger. This is that RNA I was talking about earlier, that we copy that information down from the DNA and we bring it outside of the nucleus. It's a disposable. It's kind of like a note. If you open up grandma's cookbook and you look at stuff, you're just going to scribble down what you need for that recipe. You're going to use it and you're going to toss it out. So that's what messenger RNA is. It's taking the information from DNA, leaving the nucleus, and then going to a ribosome. The second type is called tRNA, or transfer RNA. It also gets its name because if you look at the structure of it, it looks like a T. Yeah, that's right. So transfer RNA, what its whole job is to transfer amino acids to the ribosome to decode the mRNA that's on the ribosome. Okay? On the tRNA, there is something called an anticodon. We're going to come back and we'll be dealing with this. We'll be modeling this. So the anticodon is going to be what's used to pair up the correct amino acid with the codon illustrated on the mRNA. Another type of RNA is the rRNA. This stands for ribosomal RNA. There are 2 major subunits of rRNA that come together. If you look at this sweet GIF right here, we have these, see, kind of like circular helices there in the orangish. That is actually the RNA. The purple represents some protein structures that help support that. But these 2 come together and they literally clamp around the mRNA. And then they wiggle back and forth. And when doing so, they are creating that polypeptide that will come out. We'll model this in class as well. So this is what it actually looks like in the real world. Here is your RNA family photo. You have your rRNA in this big red ribosome. You have your mRNA carrying the message from DNA. And then you have your tRNA coming in. And you have the codon on the mRNA and the anticodon on the tRNA. What that is doing is decoding the message from DNA and making an appropriate, a complementary sequence of amino acids. Those amino acids, the order of those amino acids is coded for in DNA. So here, this is the big picture where we're going. Remember, DNA is transcribed into mRNA, and then that mRNA is translated into a protein. All right, that's all I've got for you today. I hope that was informative. Here is your moment of monkey and your question. Enjoy.